Hi guys, hope you will be fine at home, good at home. Today we will be starting with the proteins and the amino acids. So the amino acid that are that amino acids that are the building blocks for the proteins, and there are numerous amino acids, and you know that the proteins that are on average providing 4 kilocalories energy per gram. So talking about the proteins, as we know that the amino acids that are the building blocks for the proteins, and there are about 20 amino acids in the protein. So look at this one. There are about 20 amino acids. So what are the basic structure of the amino acid? So each amino acid that comprises of a carboxylic acid group and amino group, and this is the called as the alpha carbon, and there is elongation of the alkyl group. So this one structure, that will be the same for all the amino acids, and there will be difference in the R. So all of the amino acid that will have, this is the basic structure, and this will be the variable R. So we can classify these amino acids into different ways. Some of the amino acids, for example, some of the amino acids that can be classified into essential and the non-essential amino acid, and these are the important classification for the especially the nutrition people. So some of the when we talked about the chemistry, food chemistry, so there is a different classification. These classification that is actually based on the polarity, side chains, charged side chain, either the charged or the uncharged side chains. And on the nutritional basis, there are the essential amino acids, there are the non-essential amino acids. So will be looking both of these classifications. So first of all, we are looking the essential amino acids and the non-essential amino acids. Essential amino acids are those amino acids that are essentially required by the human body but not produced by the human body. Then non-essential amino acids, these are normally produced by the human body. So what are these essential amino acids? Valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, threonine, histidine. Histidine is especially considered as the essential for the infants only. Then lysine and arginine, these are called as the semi-essential amino acids. And among the non-essential amino acids that are produced by our body, these are the glycine, alanine, proline, serine, cysteine, tyrosine, asparagine, glutamine, aspartic acid, glutamic acid. So we will be looking the structures, chemistry structures of all of these amino acids. And another classification, normally the food chemist that is normally using this type of the classification, that is based on the charged and uncharged and the polarity. So amino acids with uncharged polar side chain, these are the serine, threonine, cysteine, tyrosine, asparagine, and glutamine. We will be looking all of these in the structures of the amino acid, chemical structure of the amino acid. Then amino acid with charged side chains. These are the aspartic acid, glutamic acid, histidine, lysine, and arginine. So these will be more clear in the structures of the amino acid. So these are the structures of the amino acids, and you will be remembering all of these structures. You will be practicing all of these structures of the amino acid in this order as I am telling you. So first of all, we will be looking, these are the further classifications. So first of all, we will be looking as hydrophobic and non-polar. You are well aware of this meaning, hydrophobic. And non-polar mean the side chain. We are talking about the side chains. So when we talk about the side chain that is not contributing to the polarity. So if we look at this one, the basic structure COO and NH3 and CHH. So if I uh, explain it further, look at this one. Just I am opening the drawing pad. So look at this one. So on one end of each amino acid there is a carboxylic end, and this is the alpha carbon, and as you know this is acidic form, and on other end there is the amino group. So this is the amino group, and this is the, you can say this is the general structure for each amino acid. And this, there is the, on this end there is the R, alkyl group, right? So the simplest amino acid, that is the actually the glycine. How can it be made? So as I told you earlier, that this is the same structure for all the amino acid. We have to change here only. So for glycine, what will be this structure? This is the structure for the glycine. COOH, CH2, NH2, right? So look at this one. COOH, NH2. This is in the hydrated form. When this is in the hydrated form, we can write it COO minus NH3 plus. So on one side H and other side another H. So this will be the glycine. It's a non-polar. So due to the presence of this H that is not contributing towards the polarity, so it's a non-polar amino acid. For the alanine, simply what you are going to do, you are just replacing this H into CH3. This structure, this will remain the same, and you will write here CH3 alkyl group, right? So what is the structure? This is the alanine. Again, it's a non-polar hydrophobic type of the amino acid. Then the valine, it's a branch chain. You can see here, it's a branch chain amino acid. This is the branching you can see here, right? So what are you doing? So CH, or you can create the branching. This is the branching. Again, another branch, and CH3. CH3 and CH3. So remember, it's a branched amino acid. This is the valine. Similar is the case with the leucine. You are adding an CH2 here at that point, CH2. So you are actually adding a CH2 here. CH2. Right? So this is again the hydrophobic and nonpolar leucine. Then same case, it's you can see here this is also a branch chain. Valine, leucine, isoleucine. These are the examples for the branch chain amino acid. Same is the case with the isoleucine. What you are doing, CH, end up creating a branch, CH3, CH2, CH3. Right? So it's a branch. So these five amino acid, glycine, alanine, valine, leucine, isoleucine, these are the hydrophobic and the nonpolar, and you have to remember these in this same order: glycine, alanine, valine, leucine, isoleucine. Right? So then there is some hydrophilic and polar uncharged, polar but uncharged. And from where this polarity that is originating, that is actually originating from hydroxyl group, this side, hydroxyl group. So there are two amino acids that contain the hydroxyl group. One is the serine and other is the threonine. These two amino acids containing the hydroxyl group. The basic structure is the same again. Again, this is the same, right? Again, this is the same structure, and what is the changes, because that contain the hydroxyl group in it. So what are you doing? You will be adding CH2OH. This is the hydroxyl group. This is an uncharged, but this is contributing actually the polarity hydroxyl group. So this is the serine. Another with the hydroxyl group is the threonine. This is the basic structure, CH2OH and CH3. So what you are adding, another CH3 here. So this CH3, CH2OH, that is contributing the polarity, but there is no charge on it. So this is the serine and the threonine. Then there are two amino acid in the nature that is containing the sulfur in it. Just we see the hydroxyl group with two amino acid. There are two amino acids, cysteine, another is the cysteine, and another is the methionine, actually the three one, that contain the sulfur into it. And again, these are the hydrophobic type and non-polar. That is not contributing the polarity. Here we are looking at that containing the polarity. But here there is no polarity. It's a non-polar amino acids. So first of all, we will be looking at the cysteine. So the basic structure is the same. This is the basic structure. CH2, and because this is a sulfur containing, so we will be adding the sulfhydryl group here, SH. So this is the amino acid cysteine. Then we will be looking at the methionine. This is the basic structure, CH2, CH2S, CH3. This is the methionine, right? So these are the example for the hydrophobic and nonpolar, especially you can remember this for the sulfur containing amino acid. Then there are two amino acids that are acidic in nature, and that have the extra charge on it. And you know that the acid behavior is due to the presence of the carboxylic acid. So these two amino acids that will be containing extra carboxylic acid into it, right? So we will be adding an extra carboxylic acid at that point, CH2 and COOH. So this is the aspartic acid, and for the glutamic acid you will be adding another CH2 at that point. So, and this is the acidic and the charge. Acidic behavior is due to the presence of this extra COOH group, and you can see the charge on it. Then against to that one, there are some derivatives from amino acids. One is made from the aspartic acid, this is the aspargine, and other is the glutamic acid, that is the glutamine. So look at this one. This carboxylic acid is actually replaced by the amide group, amide group. So what is the amide group? So properties has been changed. So what is the amide group? C double bond O NH2. So this is the amide group. So it will be now called as the glutamine, that is made from the glutamic acid. So it's called as the glutamine. Same is the case with the aspargine. Sorry, it was the aspargine that is made from the aspartic acid, and same is the case with the glutamine. Just like that, CH2, CH2, COO, this is replaced by the amide, CO and H2. So these are the acidic one, and these containing the amide group with it. Then we will be looking at the basic amino acid that were previously we have seen the aspartic acid and the glutamic acid. These have the acidic behavior, and these three amino acids that have the alkaline behavior, lysine, arginine, and histidine, right? So that is containing an extra amino group in it. That is containing extra amino group in it, right? So what is this one? This is the same structure as we discussed, and there are 4 CH2 and 1 terminal NH3 in the lysine. And arginine, there are 3 CH2, then NH, C double bond NH2 and NH3. And for the histidine, this is the same structure, C double bond O CH NH3. And this is the actually containing the amino group, additional amino group. You can see here. So you can draw again. So you can draw this one, and then here the CH2, here's the amino group here. So look at this one, CH2NH, NH, right? So this is the example for histidine. Then, as you know, that is the basic amino acid, and this is the reason for this some extra amino group. That is the reason for the base alkalinity or the basicity. Then there are two amino acids. One is the phenylalanine and other is the tyrosine. Again, these are the polar type of the amino acid, and these are somewhat aromatic. So first of all, we are looking at the phenylalanine. So this is aromatic, right? So this is the actually phenylalanine. This is hydrophobic and non-polar. For tyrosine, just add OH at that point, right? So these are the aromatic type, and these are the non-polar type of the amino acid. And finally, other aromatic class, these are the hydrophobic and the non-polar. There are two amino acids, proline, and other is a tryptophan. So in this case, this is a unique, you have to make the 5 corners for this proline. On one end COOH and on other end NH2. In the case of the tryptophan, this is the same structure, and you have to make a double rings here, NH, CH, double bond C, and CH2 with that structure. This is the tryptophan. So that's all about the structures of the amino acids. So this page also show the structures, but additional thing that is the amino acid, each of the amino acid, you know that each of the amino acid that have a specific name, and that can be either coded with 3 letters or single letter. This is the nomenclature for the amino acid that can be denoted by 3 letters or a single letter. So for alanine, ALA, or you can denote it as capital A. We have seen the structures as well as its nature, neutral, nonpolar. Arginine that can be represented by ARG or R, asparagine, ASN, and N, aspartic acid, ASP or D, cysteine, CYS, or capital C, glutamic acid, GLU, or capital E, glutamine, GLN, or Q, glycine, GLY, or capital G, histidine, HIS, or capital H, isoleucine, ILE, or capital I, leucine, LEU, or capital L, lysine, LYS, or capital K, methionine, MET, or capital M, phenylalanine, PHE, or F, proline, PRO, or capital P, serine, SCR, or capital S, threonine, THR, or capital T, tryptophan, TRP, or W, tyrosine, TYR, or Y, valine, VAL, or V. So these are the nomenclatures for various amino acids. So we have seen the amino acid that can be differentiated on the basis of the polarity and its acidic, alkaline or the neutral behavior. We have seen non-polar and neutral, we have seen polar and neutral, we have seen acidic and polar, we have seen basic and the polar amino acids. So what is the reason for these? What are the basis for these polarity? Basically, the greater the electronegativity difference between atoms in a bond, the more polar the bond will be. And partial negative charges are found on most electronegative atoms. The others are partially positive, right? So nonpolar behave, we have seen some of the nonpolar amino acids because the side chain which have pure hydrocarbons are the alkyl groups, are the aromatic groups. These are nonpolars. For example, valine, alanine, leucine, isoleucine, phenylalanine. And the number of alkyl group actually influence the polarity. The more the alkyl group present, the more non-polar the amino acid will be. And among the side chain polarity that actually differ based on the side chains. What we have seen the acidic nature of the side chain, amide nature of the side chain, alcohol OH, just the serine, alcohol OH, and the amines that will impart a more polar character to the amino acid. And the ranking of the polarity will depend on the relative ranking of the polarity of different functional groups. For example, if we look at the aspartic acid, that will be more polar than the serine because acidic functional group is more polar than the alcohol group. So look at this one structure. We have this one, this is the polar. Serine is also a polar. Here the polarity is due to the OH. Here is the polarity is due to the COO, right? So both are the polar type of the molecules, amino acids. So comparing the polarity of this one, this is imparting more polar nature, COO. This is imparting more polar nature as compared to this one, OH. This is an alcohol group, this is an acid group. So acid group has more polarity as compared to alcohol group. Both have the polar ends, but the aspartic acid that will be more polarity as compared to serine or the threonine. So serine is more polar than the tyrosine. The reason is since tyrosine has the hydrocarbon benzene ring on it. So these are the some examples why the polarity is there with the amino acid and what will be the higher polarity associated with the amino acid and where the that is the lower polarity. On the basis of that, some of the amino acids that are acidic side chain, basic side chain, that some of them have the neutral side chains. So we will be closing our lecture here.